In this video, we're going to look at the cell cycle and controls on cell growth. In an earlier discussion, we learned that all cells come from pre-existing cells, as we discussed the cell theory. Here I have a rather crudely drawn cell, but we can see a cell with a nucleus and inside 4 chromosomes. So if this cell were to give rise to these two cells, let's discuss what events have to happen. First, let's look at just the genetic material. On this side I have 4 chromosomes total, and over here I have 8 chromosomes. So somewhere during this process I'm going to have to double the amount of genetic material, or we're going to have to replicate our DNA. If we look at just the nucleus, on this side I have 1 nucleus, and on this side I have 2 nuclei. So we're also going to have to double the number of nuclei, or have nuclear division, in this case mitosis. And then look at the cell membrane. Over here I have 1 cell membrane, or 1 cell, and over here I have 2. So we're also going to have to double our number of cells, or have cellular division, or cytokinesis. So we need to think about all 3 of those steps having to occur to go from 1 cell to 2. Let's look at where each of those events happens in what we call the cell cycle. When a cell is created, it enters into what we call the G1, or the gap 1 phase, sometimes called the first growth phase. This is basically the day-to-day life of a cell, where it's going along doing whatever that cell is designed to do. For many cells, they're never going to leave G1, and they move into what we call G0, or a stage of life where they're never going to divide. They're going to do their job until it's time for that cell to be gone or to die, but they're never going to divide again. Brain cells tend to do this, neurons. But other cells are going to grow, and sometime in their growth and in their day-to-day life, they receive some signal or stimulus that it's time to divide. And they enter into what we call the S phase. And S stands for synthesis. It's during the S stage when DNA replication occurs. Now in this video we're just going to say DNA replication occurs, but for a more detailed discussion on how DNA replication occurs, click on the link that I'm going to have right here. It'll take you to a video on DNA replication. But after we replicate our DNA and we double the amount of genetic material, it's time for G2, or gap 2. And this is a time when a cell starts stockpiling raw materials in preparation for this division. Now that we have all the materials we need to divide and we've doubled the amount of genetic material, it's time for mitosis, or nuclear division, where we divide up this genetic material into 2 equal nuclei. And most of the time, following mitosis, we have cytokinesis, or cell division. Now, in the analogy we've made throughout the course and in the class, we talked about the cell being a factory. In that case, G1 would be the day-to-day life of that factory, producing whatever that factory does. And at some point, decisions made that it's time to split that into 2 factories, and then the first event we'd want to do is double the amount of instruction manuals that we have. So we have DNA replication, and then gather up all the raw materials, the extra machinery, the parts we'd need to start up 2 factories, and then divide the 2 copies of the instruction manuals into 2 central offices, and then of course divide into 2 factories. Now if we look at the cell cycle in a slightly different way, we can see that G1, S, and G2 are actually part of what we call interphase, and the prefix inter means between. So interphase is the time in a cell's life between divisions. So G1, S, and G2 are the 3 stages of interphase. And after interphase we'd move into mitosis. We have the stages of mitosis: prophase, metaphase, anaphase, telophase, as the stages of nuclear division. And in this video we're not going to go into the details of that, but in the video that follows this one you'll see all the details of how this process occurs, and also cytokinesis, or cellular division, is in the video that follows this one. So watch that one for the details of that process. In this video though, we're going to focus more on interphase and the time that we spend in interphase, and what kind of controls the passage from one part of interphase into the other. Basically, this is the lifespan of a cell, the time that it spends doing its job in between divisions. And this cell lifespan can be influenced in terms of its length by maybe the type of organism that it's in, the type of cell within that organism, or even the age of that organism. So let's look at some of the factors that regulate cell growth in healthy cells. Recall from one of our earlier videos when we talked about the parts of the cell, we discussed a problem about and asked a question about whether cells can be large, and we said that we had what we called the surface area to volume ratio problem. That as a cell grows larger, its needs increase, as do its wastes. And so cells work really hard to maintain a balance. We call this homeostasis, the movement of materials in, nutrients in, and waste products out. And all these materials in and out have to come in across the plasma membrane. And the larger a cell gets, the harder it becomes to maintain this balance, this handling this movement of materials in and out of the cell. As a cell grows larger, its volume will grow faster than its surface area because volume is a cubic function and surface area is a squared function. And as the volume increases, the needs increase. We have to move more things in and more things out of the cell. But these needs have to travel across the surface area to get in. There's more cellular traffic, but not enough roadway to handle it. We said this is one of the reasons that cells are limited in size, or at least depending on their shape. So the key issue is the surface area to volume ratio. We'd like to have a large surface area compared to the volume in order to make it easier to maintain homeostasis. So any cell that's gotten too large and it's struggling to maintain homeostasis, mitosis and cell division would be a good solution. In fact, this increasing difficulty in this maintenance of homeostatic balance can be one of the signals for mitosis and cell division. Another type of growth regulator are chemical signals. And there are a lot of these chemical signals. I'm just going to highlight one briefly here, and that's a molecule or a chemical called chelone. This is a glycoprotein that's produced by the cell itself, and it will inhibit mitosis in those cells. So it's a molecule or a chemical that, if absent, will trigger mitosis and cell division, but if it's present, mitosis and cell division are inhibited. And there are also outside chemical signals that can stimulate growth of cells, like hormones. A third type of growth regulator is a concept called contact inhibition. So if I have a culture medium here and I put some cells in there, those cells will start to grow and divide and start to fill up this space. Eventually, these cells that are growing and dividing will kind of cover the entire surface of this medium, and then the interesting thing happens. They stop dividing. It's as if they understand that they're out of room. The physical contact on all sides by other cells inhibits any more cell division. And we know this because if we take a group of these cells out, like this, move these out, the cells along this exposed region will start to divide again until they fill that space, and then they'll stop. So what's going on in all these cases? Well, this is a diagram with a little bit more detail than most of us need. If you're in my advanced placement class, we'll talk about this in a little bit more detail, but basically what we're seeing here is that there are 3 checkpoints that are in this control system. Whoops, I did not mean to move that. Put that back. One of those checkpoints is in G1 in the first gap phase, and then basically a cell will stay in G1 until we trigger that checkpoint, and then a cell will start and go through all the way through S phase and into G2 until it reaches its second checkpoint, and it'll stay in G2 until it gets a signal to move into mitosis. But then there's a third checkpoint in metaphase where a cell will start mitosis and stop before anaphase until it gets a signal, and once it gets a signal to go through anaphase, it will complete its mitosis, and then usually again followed by cytokinesis. So all those examples of growth regulators we talked about before are really influencing this pretty complicated system. And again, we'll talk about this more in AP class, but probably not in college prep or honors. And while all that's interesting, what's more interesting is what happens when cells lose their ability to respond to the factors that regulate their growth. They lose their ability to recognize their growth regulators. They start to grow and divide out of control. And as a group of cells continues to grow unchecked, it becomes what we call a tumor. And we can categorize tumors as benign or malignant. And benign tumors are non-life-threatening. They don't invade neighboring tissues, though if they're they can become painful. They can press on other organs and cause pain, and certainly cause problems that way. Oftentimes they can be pretty easily removed. An example of a very simple benign tumor are simply something as simple as a wart. But malignant tumors are life-threatening. because they invade neighboring tissue and can metastasize and actually cause the neighboring tissue to also become tumorous. They can break off and spread through our bloodstream and spread. They can be removed surgically, but can reoccur, and it's hard to get all of them out sometimes. And they also can be treated with drugs, chemotherapy, and radiation with varied success. And if it sounds like I'm talking about cancer here, it's because I am. These malignant tumors are cancer. We call cancer a condition where these malignant tumors will invade and destroy body tissue, and the key symptom or the signified by very fast mitosis and cell division, basically unchecked growth. In these cases, these cells have lost their ability to recognize the signals that tell them to not divide. It's interesting that we have genes we call oncogenes, which are genes that can cause cancer. But many people will carry these genes, but usually they're switched in the off position and are inactive unless they're altered or influenced. And some of the things that can alter or influence these genes and cause cancer are we call carcinogens. And they can be things like radiation, like X-rays, and even exposure to sunlight. Viruses we know can be cancer-causing. They can switch on oncogenes, and certainly many various chemicals can act to be carcinogens. They attack and damage the DNA of the cell, which disrupts this control of cellular division and mitosis. And this is a picture of cancerous tumors or tumors on a cancerous liver. And that brings us to the end of our video on the cell cycle and controls on cell growth. Click down here on our next video where we'll go into the details on mitosis and cytokinesis.